In my today's lecture, I'm going to talk to you about some of the basics of cell and then move on to the central dogma of life. So if you think about the origin of cell, we have to think about a time scale of billions of years, right? So we are really talking about something to the tune of 4 to 5 billion time scale when during those time, how you can see like, around 3.5 billion years ago, microorganisms started growing. And then around 2.2 billion or so, some of the cells with the nuclei, which is having nucleus, they started, appearing. And then the atmosphere was changing with more oxygen forming atmosphere was conducive for the cells to grow. And more life evolved from that time onwards. And then more of the, so these are more microscopic cells and organisms. And then we got more macroscopic organism in the 0.6 or so billion years ago, from where then different taxonomy of animals started coming forward, including dinosaurs and all, which have got good attention from the all our frictions, and then of course human being, which is still not so long if you see the trajectory of billions of years scale. So just to relate to you, what we are talking is cells. And if you think about, the atoms and molecules, now then these cells, these are the basic structural and functional units of life. And these cells, which is what we are constituted of these cells, which is almost billions of cells, which makes us, right? And these cells, they are forming the tissues. So when we say that, muscle tissue, or, you think about any other tissue of the body, those are composed of very specialized cells. So now these cells are forming the tissues, and different tissues are going to form the organs. And then those organs are making, an organ system followed by an organism. So now I hope this gives you the bigger picture. What we are talking is really microscopic and very small scale when we talk about cell and molecules. Uh, but what you most of the time think about biology essentially is, when we talk about a disease, somebody got a heart disease, somebody, got a liver disease or renal failure, those kind of problems. But then they all go back from which tissue they are composed of, which cells they are composed of, what happens in the cell at their DNA, RNA, and protein levels at the molecular level. So again, to give you the perspective, when we talk about human body made of billions of cells, every cell looks like this, where the nucleus part of that, this is the nucleus shows, contains the chromosomes, from where then we have the nuclear material, and we have the genes and the DNA. If you think about analogy with the, a complex model of a car, when you open all the nut and bolts of the car, and now if you think about a similar way on the left side, the nut and bolts of the cell, the circuits looks very similar. In fact, there are labs, the Shapiro lab at Stanford and other groups are working. They're trying to make, electric circuit of the cell and trying to connect all the organelle. That's how they're all working with the master as the nucleus and how they're all interconnected, right? So now I think you got some idea that what we are talking is really minuscule, and to work, the life system to be functional, there are many things which has to work in conjunction, and all of those only together constitute the life. So this is really, on one hand, complex, but on other hand, very interesting, because you can start thinking about complex circuits and how best now you can extrapolate that knowledge here. So these cells are broadly, grouped into two different groups. One is prokaryotic cell, other is eukaryotic cell. The terminology of prokaryote, pro means primitive, very old, before, and U is the true. Carry-on term comes as a nucleus. So if we have very primitive nucleus, that is the prokaryotes. When we have very well-defined nucleus with the nuclear membrane, that is known as eukaryotes. So all the microorganisms like bacteria, viruses, other small microscopic organisms which we talk, those are essentially some of the prokaryotes. And as you can see, one of the bacterial cell structure here, it shows that the nucleus is very primitive. It means it is actually not having a well-defined nuclear membrane, and the nuclear contents, especially the even all the ribosomes and nucleoid, bacterial chromosome, they're all floating in the cytoplasm of the cell. Think about now a balloon, which is the cell, and within that balloon, everything is floating. There is no distinct another small balloon which makes nucleus. So whereas in the eukaryotes, which is both animals and plants, in both of them there is a very well defined nucleus. So there is a nuclear membrane and the nucleus. So now the same balloon which we are talking, the bigger balloon, has a smaller balloon as well. And that smaller balloon has a boundary which is defining as a nuclear membrane. So now you think about that big balloon and the small balloon inside. In between the two, there is a liquid there, and that liquid is cytoplasm. So nucleus, the center part of that small balloon, that contains all the genetic information, the DNA, RNA, everything is there, and that all the chromosome nuclear material is there. And the outer layer, the balloon's outer layer, that is what is shown here, which you see this boundary here, and the nuclear membrane boundary is shown here, right? So the outer membrane boundary, that is known as plasma membranes, and inner membrane boundary is known as nuclear membrane. In between the two, there is a cytoplasm, which is something like gluey material, semi-solid, which is proteinaceous in nature. And in between these two only, all other organelles are placed and not fixed. They're all floating, right? So you got, now I hope you can visualize that in between these two balloon layers, you got some semi-solid material, which is viscous in nature, and that material has all these organelles. And that's what these organelles like Golgi apparatus, mitochondria, peroxisomes, cytoskeleton material. So I'm not going too much detail about all of these organelles, but these are various organelles which are essential to function for the cell, right? I will talk about some of the role of these, but just right now giving you a macro picture about how a eukaryotic cell looks like. A similar concept is also in the plant cell. When you have a defined nucleus, you also have now the membrane, but in addition to that, plants have a much thicker cell wall as well, because they have to be exposed in the sunlight and in the outer atmosphere. So they cannot cope up with that. So then they also have a cell wall. Now, in between these two of the bigger balloon, which contains now cell wall and plasma membrane, and a smaller balloon, which is the nucleus, now you have all the organelle which is floating around. And one of the organelle which takes very long space, that is the central vacuole. So that is actually a very distinct part of the plant cell. So most of the undigested product and the waste product and toxic material, they all come and get stored in the central vacuole in the plant. Now, again, in between the two, we have also some other smaller organelle, which is very important for cell to function. So some of these concepts, could be very basic for some of you who are already coming from a little bit bio background, but I think it will be a good idea for us to have the whole class, whole group to be on the same page. So let me now take some of these organelle and define to you what their functions are, right? So cytoplasm, as I mentioned, in between the smaller balloon and the bigger balloon, you got viscous material, which is refers as a cytosol or cytoplasm, and that contains some of the protein complexes, which along with the ribosomes and cytoskeleton material. Now, cytoskeleton, the way we have our skeleton, which makes us to stand and which makes us to position properly, like that, cell also want to have a proper shape. So for the proper positioning of the organelles, the cell contains certain cytoskeleton material, and those provide help to not only position organelle, but also help to transport the material from one cell to other cell and the movement of the chromosome. So these are known as cytoskeleton material. Vacuole, as I mentioned to you, that is the place where all the the waste products can be accumulated, and that's more exclusive for the plants. So now, just to give you slightly more detail about each of these organelle, which I spoke to you, and now again also refreshing about the basic broad overview. We have a nucleus in the eukaryotic cell. Eukaryotes means we are talking about more evolved nucleus. Prokaryote, more primitive nucleus. In prokaryote, we don't have this distinct nuclear membrane. And now we got two different membranes. One is plasma membrane, but one is nuclear membrane. So the plasma membrane is, which is the outer layer. So now think about the bigger picture of the human body. And as I said, we are talking about now not only 1, 200,000, 1 lakh, 10 lakhs, but we are talking about potentially millions to billions of cells, right? So they're all, squeezed into this small microscopic, the dots, which is all in our body. So they are the one which are actually making all the communication from one cell to other cell. A heart has to really work well, and for that, a lot of signal has to come from brain, and lot of information has to move from one cell to other cell. So there has to have some communication, right? So intra-communication and intercommunication, all of those are possible because of these plasma membranes, which is providing that boundary, and also they are helping for transport of the molecules, like and the receipt of the cell signals. All of those are being governed from this small plasma membrane, which is people have looked at in the electron microscope and try to define their structure and how best they are able to communicate these signals. Throughout, actually, I have defined some figure numbers. These are from a textbook, Campbell Biology, which we take as a reference book for teaching at IIT Bombay for our undergrads. I'm sure many of you who are intended to teach to your UGs, I think this is a good book to refer to. And most of the figures which I have referred numbers here are coming from the Campbell Biology. So as I mentioned that the nucleus is the one which is defining the definition of eukaryotic versus prokaryotic, and that is most prominent organelle in a given eukaryotic cell. As you can see, it takes most of the space. It contains the most crucial information for this cell, which is the genetic information, and that's where the DNA is located. And from DNA, now the replication process will happen, and then that is going to form the RNA from that. From the RNA, now the protein synthesis will happen, and that's where then the proteins will be generated, and they will be moving out from there to the main cell. So another organelle which is of interest is endoplasmic reticulum, which you can see like this kind of membranous structure. So now you got a nucleus which is having the nuclear material like DNA and RNA, and from there the ribosomes are going to synthesize the proteins of interest, uh, and this entire system is governed through some membranes, and it is like the FedEx system where you can think about how the material has to be distributed. Everything has to be very well defined beforehand, and then only when a big shipment comes from the plane, it will go to different trucks. They will take for different locations. Then material will come very accurately and very quickly to the right spot. So like that, endoplasmic reticulum has their own information network, which is this kind of endomembranous system. And they are two types. One, sometimes these membranes you will see dotted membrane, and that's where another organelle is placed that is known as ribosome, which is the organelle which is required for the protein synthesis, where the proteins are formed. So when we have the rough endoplasmic reticulum, which is what you see it here, that is where on the endoplasmic reticulum membrane we have the ribosomes, and that is going to do the protein synthesis. When some of them are a smooth part, then that's where there is no ribosome is there. So then that will not be involved in the protein synthesis. But it is still important to make that membranous system so that the information can be commuting from the nucleus to the RER to the SER to the Golgi complexes, another organelle, Golgi vesicle. So as you can see now, this membranous system, there are also some isolated membranous structures, and those are more which is involved because the protein has to now move from this cell outside. So now there has to be some sort of post office type of content here. So that's process known as exocytosis. Please do not worry about terminology. Exo means going out, cytosis means of the cell. So now Golgi vesicle will help these proteins to transport from one cell to other cell. So now another organelle which is more specialized, that is peroxisome, which contains some specific enzyme like catalase enzyme, and that helps to do the fatty acid molecule breakdown. And this organelle is important for various type of lipid and fat, those kind of degradation. Whereas it does not contain any other specific DNA or its own molecule. I'm just giving you one term, like ideally one would expect that only nucleus contains DNA in the cell, right? But then we also have another organelle, which is mitochondria. Even mitochondria contains some DNA, whereas the peroxisome and other organelles, they do not contain DNA. So mitochondria, it is one of the very specialized organelle in our cell. So if you have to remember at least two organelle name from the given cell, at least you should know nucleus. Nucleus contains all the genetic material required, which is the DNA and RNA. And now another organelle, which is mitochondria. Mitochondria is the powerhouse of the cell. All the energy which we want to use, which is being generated, it all comes from the mitochondria. So mitochondria is the place when all the oxidative metabolism happens. It means, all the metabolic activity which we are doing, for that ATP is synthesized from these mitochondria. And mitochondria is very specialized organelle because it contains its own DNA as well, which is also a peculiar characteristic. It contains two membrane structures. So every other organelle is having only one membrane, whether you talk about ER or endoplasmic reticulum or peroxisomes, lysosomes. Any of those organelles have only single membrane structure. In case of mitochondria, it has double membrane structure. Now let's go back one step and think about the evolution of life, right? And how in the ocean and those water environment, how this cell would have evolved. Still, we have no clarity that why this mitochondria is the only organelle which contains double membrane structure in the animal cell. In the plant cell, we also have additional organelle, which is the chloroplast. Even that also contains two membranes and its own DNA. So these are the only two peculiar organelle, mitochondria and chloroplast, which contains their own genetic material and double membrane structure. Whereas the rest of the other organelle is a single membrane structure and they do not have their own DNA. So one possibility which people have tried to project as hypothesis, and some experiments have been done as well, to think about at the time of evolution, when there were different cells were growing, it is possible that one of the bacterial cell got engulfed in this particular eukaryotic cell, and that particular exposure, that bacteria which came, which was of course having its own DNA and having the single membrane structure, that was trapped with this particular organelle, mitochondria, and that's how mitochondria evolved to have double membranes and its own DNA. And a similar thing is thought about for the plant cell of the chloroplast, which is another bacteria, which is cyanobacteria, probably got engulfed. And that's how these two organelles are more specialized. They can generate energy, they are double membranous, and they have their own DNA. So I think some of these things are important to keep in mind, even if it may not be subject of your specialized domain. But still, at least I hope you are able to get the broader picture about how our body is made of billions of cells, how one cell is made of the nucleus as a central component, and then many other small important organelles are there. Within those, mitochondria is another very crucial organelle which provides energy for all these activities. So just broadly, you don't have to recall all the differences, but when we are talking about plant cell versus animal cell, there are some specialized differences, like plants contain plastids, which is gives them the green color and photosynthesis ability that is absent in the animal cells. Plant also contains a large vacuole, which I showed you in the beginning, which is absent in the animal cell or very rudimentary small. A cell wall is present in the plant cell, which makes them much more robust and provides them more strength, which is absent in the animal cells. And then there are some more specialized organelles like lysosome present here. Lysosome is present in animal cell. Centrosome is present in the animal cell. So again, some of these terms you don't have to worry. I'm just giving you, for your curiosity, how these animal cell and plant cell looks in the microscope. When we do these experiments in the cell biology labs and make those microscopic slides, when you see that under microscope, how these cells look like. So let me just show you this small clip of animal cell.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
All right, so as you can see in the short clip that how one animal cell is dividing and forming two different daughter cells. I will talk a little bit about in cell division, but at least you got to see the way I showed you fancy pictures. In reality, these things does not look like that. Those are all animated.
Differential interference contrast.
And let me now take you to the plant cells.
As this plant cell, taken from a lily, prepares for division, the chromosomes first condense. Next, the mitotic spindle lines them up in the center of the cell. At the metaphase to anaphase transition, the sister chromatids of every chromosome pair separate suddenly in striking synchrony. The chromosomes are pulled along the microtubules of the spindle to opposite ends of the cell. After chromosome separation, membrane vesicles line up in the center and fuse with each other to form the new plasma membranes that separate the two daughter cells. At telophase, the chromosomes decondense in the newly formed nuclei.
All right, so from both plant and animal cell, you got some idea that how they are visualized in the microscope. And also that brings us to the next topic, which is the cell cycle, how the cells are going to get divided and forming new cells. So what we talked so far is about the bigger picture of the microscopic scale, looking at from the life evolution, moving to a cell, and then from a given cell that how different organelles are important to make that cell, which is the basic unit of life, both structural and the functional level. And now we are looking at how that one cell is undergoing the division, and for that different activities which are required, which all makes under the cell cycle. So there are three, so broadly the whole cell cycle phase is having two different major events. One is known as interphase, other is known as mitotic phase. So mitotic phase is where the division is going to happen, which you just saw in the video, that how the cells are going to divide. That will only happen here in this part. But this division to happen accurately, the cell need to be well prepared, right? And that preparation part is known as interphase, which is constituted of three different smaller events, which is G1, S, and G2 phase. All these three are very crucial for a cell to get prepared that a division has to happen. Because for division to happen, now in the cell you need to have sufficient amount of nucleic acid, you need to have sufficient amount of all the molecules which is going to make the distribution equal to two different daughter cells, right? So that's where there are different type of even checkpoints involved to make sure that the cell division which is going to happen properly. And think about, this process we are talking about, so many cells in the body, they're all dividing, and the division has to be exactly equal, so that they can replenish whatever wear and tear we have. We get sometime wound, sometime other kind of aging effect, many problems. All of those cells has to replenish, and they have to be exactly same. If they're not same, and sometime if they lose control, then that may result into disease like cancer, right? So the process has to be very much orchestrated and very regulated. So mitotic phase is going to alternate with the interphase or the growth period. And now we have three different phases. G1 phase is important as the first phase of the interphase. S phase is where all the nuclear material, the chromosomes, they get condensed and they get duplicated. And G2, this is the last phase of the interphase. So again, this is the place when the cell starts preparing for the division to happen. Now it makes sufficient amount of chromatin and the chromosome material so that the cell has sufficient nuclear material to get divided into two parts. And then last part is ensuring that now we are really ready to go for the mitotic phase. So these things, and then now, of course, at the end, when the mitosis starts, it's going to distribute equally all the chromosomes, which contains nuclear material, to the daughter nuclei. After the, the chromosome distribution has happened and division has happened, then you also want the cytoplasm to be equally distributed, because as you understand now in the given cell, you have the nucleus, nuclear material, And you also have a cytoplasm, which is the semi-solid part where all the other organelles are there. So then all this entire activity has to be equally divided. So then cytokinesis is a process where the cytoplasm is going to get divided. So broadly, we have two different type of cell division. One is mitosis, other is meiosis. In mitosis, essentially that happens in all the normal body cells, which is in the somatic cells. And here you want to have the same amount of DNA to be continuously produced from the parent cell to the daughter cell, right? Whereas in the meiosis, that happens only in the gamete cell, which is a sperm and the ovum. And here you are going to produce four daughter cells, which is going to have half the amount of DNA. Because then during the fertilization process, now half the amount of DNA from the sperm, half from the ovum is going to come together, and then only the ploidy or the overall genetic material is going to be maintained. So broadly, what we are talking is something very crucial. The way we are going for the repair and maintenance of our vehicle and different other electrical appliances, it's like our body needs all the time cell to divide properly for the growth and repair, and of course for the differentiation of different specialized type of cells. So these are very crucial process which happens all the time, and the if you think about significance of that is very much important when the new life forms starts, where now the sperm cell and the ova from which the egg cells are produced, they are going to fertilize, and then they are going to form an embryo from which multiple stage of embryo development will happen, and then that is going to continue to evolve into the life form and the baby is born. So the cell division is going to distribute the DNA, which the chromosome, which is contains DNA of the mother cell equally into two daughter cells finally, and then it allows the cell to proliferate. Uh, I'm sure you are aware now that these cell divisions are so crucial that if they are not happening properly, all our activity of the body cannot perform properly. So a little more technical terms here, but I think you should be slightly aware of that the somatic cells are essentially the normal body cells, like a skin cell if I talk, or other hair cell and all other normal body cells, which are non-reproductive, and they contain two set of chromosomes, and they're continuously dividing, and the DNA contents and their ploidy is maintained. Whereas the gamete cells, which is in the male the sperm cells and the female egg cells, they are having half as many chromosomes as the somatic cells. So now this is a broad picture of the reproduction process, where meiosis is very important event, which is the green circles are showing, the green arrows are showing here haploid level. It means the genetic material is half, and the orange ones is showing the diploid level, which is 2n. It's double. So now, if we look at a normal cell, which is the diploid zygote cell is formed, which is having 2n equals to 46 chromosomes. So now this 46 number is going to continue in the normal cell for the mitosis and development to happen. And that is what is going to make the normal body growth and repair. Whereas specialized cells from the ovary and testis, they are going to undergo the meiosis process, which is going to reduce the ploidy level of these chromosomes. And now we got 23 haploid gametes from the egg and same from the sperm. And both of them after fertilization is forming 2n equals to 46. So in the meiotic process, the ploidy is maintained from one generation to the next generation, so that the overall chromosome as well as the entire genetic informations remains the same. This is very specialized cells which is performing this process of meiosis. Rest of the body cells, they are performing mitosis process for the routine body's requirement and growth and repair and differentiation process. So now if this particular cell cycle which we are talking, if it is happening properly, everything goes well, but sometime it may not happen properly, and that dysregulation could be very costly for us. And that's where many complex disease like cancer, which is happening, is because of this cell cycle dysregulation, right? So as I was talking to you when I talked about the cell cycle overview and the cell division, we got two different major processes. One is the mitotic phase where the cell is going to divide, and remaining is all interphase, right? So interphase is what is going to prepare the cell for. It's like when you are just going, in a car, very, with a high speed on a highway. So then you will see all the time there is, some sort of brakes are there, speed breakers are maintained so that you can reduce your speed and you are going properly with a static speed. Same phenomenon here. Now in the G1 phase, now you got a G1 checkpoint. Now that is ensuring that now cell has all the nutrients and material to continue the division process to happen. So now cell is ready to move to the S phase. S phase is a very crucial phase where all the nuclear material which contains chromosomes and DNA, they are going to synthesize, chromatin is going to synthesize, and it's going to really get condensed. So that's where the replication process happens. And but before replication happens, the cell has already ensured that now good to go from G1 to the S phase. Now when S phase has happened, after that, if every material that was condensed is that sufficiently produced and can move from the S phase to G2 phase, that is being ensured by the G2 checkpoint. So we got two different checkpoints. One was G1 checkpoint, other is G2 checkpoint, and these two checkpoints are nothing else but some proteins and some receptors. So I'm not going in too much detail, giving the the background may not be required so much to go in detail. But here we are not talking about a physical checkpoint. Here there are some protein or receptors which are present, and these are known as cyclins. And now on those there is cyclin dependent kinases which is coming and binding, and those are the one which is ensuring that now cell is sufficient to move from the one phase to other phase. So I'm just trying to give you the flavor and the bigger picture of how the cell operates, how the cell divides, and what is the at the molecular scale, how it is being maintained well, right? So once it has reached to the G2 phase, then now cell is moving towards mitotic phase, which is where the division is going to happen. And again, after mitosis completes, there's another checkpoint, M checkpoint, which ensures now all the the DNA material has now divided equally into two cells. Cytoplasm, cytokinesis has happened completely, and now one cell has formed two cells, which is identical and contains same information. So in addition to these internal cues, there are also several external things which is equally important. For example, whether nutrient is sufficient in the cells, growth factors are there. Also, space is very important, and that's where a lot of engineering principle comes into the play. There are many mechanical engineers and people working in the from engineering background. They try to study the cell cycle and the cancer, looking at different properties of mechanical experiments. For example, like we are talking about our body, defined body shape, and within that body we have billions of cells growing. And now if in that space, if some cells start growing very rapidly, like every cell was only growing when there is a need, if some cell got damaged, some cell got you lost some from an accident, and then you are having more doubling of those cells and repairing that, and further division is happening. But now some cells automatically lose control and then they start dividing very rapidly, and this is what is going to cause cancer. So now they are on one hand the space is limited, right? You think about a box, and within that box cells are growing and it's already filled. But then there are some cell which has started growing really rapidly, and now they are really not willing to follow the rule of that you have to now stop division because the space is limited. So then they are not affected with the density inhibition, but in general, the normal cell will stop further dividing. If there is a space limitation, they will stop dividing. And so therefore, they're also governed with the density of the space what is required. Also, the cells, when they have to do division process, they are going to get anchored on certain substrates. These substrates are some specific material in the cell which is going to help them to get adhered, and from there they can quickly move on for the division. So that is known as anchorage dependence of the substratum for the this process to happen. But the cancer cell, they are having certain inherent characteristic which they're not going to follow any specific rules of the normal cell cycle division, right? So whatever the checkpoints which we talked, the G1 phase checkpoint, G2 phase checkpoint, M checkpoint, all those cancer cells are not going to obey. They will just rapidly move and do the their immediate lot of division will happen, and that's where a lot of mutations you see in the cell coming from those DNA, because they are not obeying the right ways of division to happen. They're also not going to obey that stop the division, because now density is not sufficient. It has reached to a saturation level, so they're still going to form the multiple layers of these cells, and that's why you see those kind of tumor mass coming from these cancer cells. Also, they can move from one place to other place, so they do not require any specific place to get adhere on. Only then the growth can happen. So then they can actually move from one to other places, and then still continue to grow. So these are the one which really makes cancer cells very specialized, and they are not obeying the normal cell cycle rules, and that's why it creates a lot of issues. And this is the image which shows that if the cell was growing normally like this, but now if the cell is growing with the cancer cell, it is forming multiple layers. It is not obeying any density rule, not obeying any cell cycle regulation rules, and forming the tumor mass. Let me just show this clip, nice video, to give you an idea about the from cell cycle to the cancer.
60,000 billion cells make up the human body. They are the units that form the tissues which make up our organs, like the liver, heart, and lungs. When the body needs it, our cells split into two and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes, some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow. This is what we call the phenomenon of angiogenesis. The tumor, however, only really becomes dangerous when the cancerous cells begin to invade through the vessels into the adjacent areas and spread to the surrounding organs. These cells can then invade other parts of the body, multiply and produce new tumors. Metastasis is the term used for this spreading process. But why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables. Today, over 25 million people in the world live with cancer. 7 million die from it each year. The disease represents the leading cause of mortality for those under 65. Lung cancer is the most widespread.
All right, so I hope that gives you the good recap of various points which we discussed so far. How
60,000
abnormal behavior in the cell and the growth could constitute the tumor. Let me just take a minute here. All right. Sometimes people are coming in between, so I have to allow them, and that creates some distractions. I hope my screen is still visible. So let me continue. Okay, so by now you got an idea that we have started from the origin of life and looked at the cell. Within the cell, there are different major component is nucleus and other organelle, which are also equally important. And then how a cell cycle operates, the proper behavior of the cell. And if that does not happen properly, then probably we will lose that, and then we will have to uh, think about how a cell could actually govern the process like cancer. So now let's zoom into one aspect, which is within the nucleus. Now we are looking at some of the central molecules like the DNA and RNA. And this is known as this process of from DNA, the mRNA formation, and from RNA, the protein formation. That is known as central dogma of life. So these processes are very crucial for the life to operate, for our systems to continue the flow of information moving from the DNA to RNA under the process of transcription, and RNA to protein in the process known as translation. So briefly about nucleic acid, which is I have been mentioning to you inside the nucleus. We have two major molecules. One is deoxyribonucleic acid or DNA. Other is ribonucleic acid or RNA. DNA, that is the major component that constitutes the genetic material located in the chromosomes. RNA is the functional copy of the DNA. So all the information from the DNA which is required for the functional activities, those being synthesized, and those comes into the RNA form, and from there RNAs are going to participate in the protein synthesis form. So the double helix DNA structure was defined by Watson and Crick. I'm sure you have seen some of these pictures and the Nobel Prizes, which got based on how the DNA structure can be deciphered. And so just looking at the nut and bolts of the DNA, so that is a nucleotide, which is a subunit of nucleic acid, consists of three different small components. One is the base, other is the sugar, and third is the phosphate groups. So these three are the one which makes all the nucleotides, which is the subunit of nucleic acids. So there are two different type of sugar, which defines the deoxyribonucleic acid or ribonucleic acid. We have different type of base, and then we have the phosphate group. So let's look at these three in slightly more detail. So these bases, there are four different type of bases which constitutes DNA, and that is known as ATGC. You must have heard these term ATGC, which constitute the DNA. A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. So now, if you look at the structure, these two, the T and C, they are having pyrimidine ring, which is the 6-membered ring with 2 nitrogen, and other two, adenine and guanine, that is having the pyrimidine ring plus imidazole ring as a part of that. That is known as purine. So other one more base, that is uracil, and that is found in the RNA. So in RNA, you don't have thymine, but rather you have uracil. That is one major difference in DNA, these bases and the RNA. Sugar, as I said already, there are two different type of sugar. The backbone remains same. You have in the ribose sugar, you have on the 2' you have a hydroxyl chain, a hydroxyl group here, and on the 2' in the deoxyribose, you have only hydrogen. So these are the key changes which brings a lot of functionality difference at the DNA and RNA level. And then of course we have this phosphate, which is going to join from one sugar of the ribose to the other sugar of ribose, or the one sugar of deoxyribose to the other sugar as well. So then now let's look at how these are going to form the units together. So when we are only having a base and a sugar, so base ATGC and sugar ribose or deoxyribose, those are going to form nucleoside. Now when a base and sugar along with phosphate is attached, that is known as nucleotide. Now these nucleotides, which means all these three together, forming this multiple of these are with bond, which is known as phosphodiester bond, that together makes the nucleic acid, right? So nucleic acid is various nucleotides like these, which is joined with the phosphodiester bonds. So this is how now the DNA structure was decoded. Watson and Crick deduced the arrangement of the two strands of DNA propose a three-dimensional model, how they are intertwined, and how the sugar phosphate that backbone lies on the outside and bases are inside of this particular double helix structure. And then there are, of course, to pair the A with T and G with C, there are specific hydrogen bonds. So there are 2 hydrogen bonds with the A and T bonding and 3 hydrogen bonds with the G and C. So now if you just recollect everything, whatever we spoke so far, to think about how DNA structure can be understood, we got the building blocks of material, which is the sugar with the phosphate groups with the bases. All that is going to form the nucleotides. These nucleotides are now arranged in the 5' to 3' or 3' to 5' that order here. A is forming the bond, two different hydrogen bonds are being associated with the thymine, and three different hydrogen bonds with guanine cytosine, G and C. Now this is how the double helix of the DNA model looks like, where you have these phosphates and sugar outside and the bases are inside. If you enlarge those, this is where you have the phosphodiester bonds, which is connecting these nucleotide units. And these are the ATGC, which is the basis. So I hope you just get a good clear view of how our DNA is constituted of these three building blocks, right? And the phosphates are providing negative charge outside, and then you have the bases which are inside. Many times some of these properties are heavily used when we talk about experiments like protein arrays and or DNA arrays. We look at the DNA properties that phosphate is going to be negatively charged. So if I take a substrate which can give the positive charge, so then that can bind the DNA material, right? So some of these basic molecular structure becomes very important even for the big omics-based experiments. Again, to give you a slightly more detailed view here, the base pairing in DNA. So adenine and thymine are joined with two different hydrogen bonds. Guanine and cytosine are joined with the three different hydrogen bonds. And we are seeing here two anti-parallel strands. So it means both the strands are moving to the 5' to 3' or 3' to 5' directions. And that is what constitutes the DNA molecule. Let me show you this short model of DNA double helix strand.
Two DNA strands intertwine to form a double helix. Each strand has a backbone composed of phosphates and sugars to which the bases are attached. The bases form the core of the double helix, while the sugar-phosphate backbones are on the outside. The two grooves between the backbones are called the major and minor groove, based on their sizes. Most protein DNA contacts are made in the major groove because the minor groove is too narrow. The DNA backbone is assembled from repeating deoxyribose sugar units that are linked through phosphate groups. Each phosphate carries a negative charge, making the entire DNA backbone highly charged and polar. A cyclic base is attached to each sugar. The bases are planar and extend out perpendicular to the path of the backbone. Pyrimidine bases are composed of one ring and purine bases of two rings. Adjacent bases are aligned so that their planar rings stack on top of one another. Base stacking contributes significantly to the stability of the double helix. In a double helix, each base on one strand is paired to a base on the other strand that lies in the same plane. In these base pairing interactions, guanine always pairs with cytosine and thymine with adenine. A GC pair is stabilized by three hydrogen bonds formed between amino and carbonyl groups that project from the bases. In contrast, an AT pair is stabilized by two hydrogen bonds. The specificity of base pairing, that is C always pairing with G and A always pairing with T, ensures that the two strands are complementary. This is important for DNA replication and transcription.
All right, so I hope this nice video gives you some visual feel, this from this book, Molecular Biology of the Cell, that how what we spoke about the double helix structure of the DNA, how that one could visualize. So this hereditary material, the DNA, what we are talking is very important because all the information from one to next generation has to pass to continue the hereditary information. So the sequences of these bases, they are going to store the genetic information, and how they are defined in which sequence, that actually determines how the RNA sequence is going to be, and then from there how the proteins are going to be synthesized. So all the structural information is very important for the function of this cell as well, right? So these sequences are not just random, they are actually very important. They're going to give rise to specific genes, and those are going to be important for the RNA and the protein information as well. So base pairing concept, you have seen already the AT and GC with different hydrogen bonds. The DNA replication, it means the process of DNA to multiply, and that, if you recall, in the cell cycle, we talked about S phase, and that is the phase where all the DNA replication process is happening, so that more and more of the DNA molecules could be generated. So now again, to bring you back where we started with, we talked about a human cell, like human body, from which we have billions of cells. A cell is enlarged to look at the nucleus, nuclear components of that. From that, the chromosomes. We have all this, genetic material now, which is stored there. Very defined units of the genes are there, and now we are looking at more of the DNA, how that is going to get packed in the cell. So for a cell now, a lot of the information which is coming from the DNA, and these DNA molecules has to be packed in that defined space. So different other proteins, like which is known as histone proteins, they, along with DNA molecule, they are forming a complex known as nucleosomes, and then they are tightly packing, like the rope which you see, which is intertwined, that is forming the chromatin. And so as you can see, there are 4 different type of histone proteins in duplicate, so 8 histone proteins. Along with DNA material, they are being packed into the chromatin-like structures. This is what is going to get duplicated and getting condensed in the S phase for the further cell division to happen, right? RNA is another molecule, ribonucleic acid, which is only one difference you have, other than the base thymine. You have the uracil base here, and the RNA template is going to be defined from the DNA template, and that's important for the gene expression to happen. So in the DNA-based microarrays, when we are talking, we are looking at the expression of the RNA, because the oligos are going to be complementary binding to the cDNA, and that's where we are going to look at functional differential expression of the given genes. Yesterday, when I talked to you about the breast cancer subtyping, those are something based on these concepts of taking the RNA from the cell, making the cDNA from that, and on the chip you have those oligos, all the genes printed, and now these are going to hybridize and make those after hybridization where they are binding, it is going to show you the fluorescence signal from the Cy3 or Cy5 dyes. There are 3 different type of RNA, messenger RNA, transfer RNA, and the ribosomal RNA. Each one of those are important, but messenger RNA is the one which is important for protein synthesis, and that's what we care the most about. So if you think about central dogma of life, which we are talking, so from DNA to protein information is passing. In between, we have the RNA, which is important step, and that's important for the gene expression to happen. I'll just quickly flash you next 2 minutes or so some of the milestone discovery which you might have heard on and off for different Nobel Prizes, that how some of these are related to the DNA. So if we start this particular trajectory of various activities happen, milestones happen, I'm sure you'd have heard about Gregor Mendel's name, who is father of genetics. In 1865, he defined the law of genetics. Then another scientist, student, he defined the linear map of genes in 1913. Watson and Crick, they defined double helical structure of the DNA in 1953. Three scientists, Nierenberg, Khurana, and Hawley, they determined the genetic code. You have heard Hargobind Khurana's name, of Indian origin scientist for Nobel Prize, 1966. Then we have the scientists Cohen and Boyer. They developed recombinant DNA technology in 1972. Sanger and Maxon and Gilbert, they developed the DNA sequencing. So you heard about DNA sequencing, the Sanger sequencing methods. Those came in 1977. The first human disease marker mapping that happened on the Huntington disease in 1983. Then we had polymerase chain reaction, a process which can take a small amount of DNA, can be amplified to make large amount of DNA, invented in 1985. You hear all kind of criminal issues and those crimes where the PCR is being used to amplify the DNA. Then the Human Genome Organization was formed in 1980 to look at all the genes which makes the human body, because so far we still did not have information for all the human genes. And then over the process, then people started learning more about cell cycle regulation, and how can we take the cell from the even normal body cell like somatic cell and make it in full organism, like the Dolly, which was cloned in 1997 by Ian Wilmot. Cloning was one of the very scientific fantasy, which was really true, where you take a cell and try to use that in a way that you can reprogram and trying to produce a full organism. And sheep Dolly was produced in Roslin Institute 1997 by Ian Wilmot. It was actually the first large animal which was cloned from the somatic cell, which is the normal body cell from the skin cell. And that was, of course, the big scientific fantasy to come true, published in Nature, and lot of highlights it got. So I'm sure you'll be happy to see those pictures. So I'm very privileged to actually have at least some interaction with Dr. James Watson. I was teaching some courses in New York in Cold Spring Harbor Laboratory for proteomics field, which I work on. And it was long interaction with him of almost 2 weeks or so. Couple of times when we went to teach the courses. It was really good to spend some time with him and share his journey of the Nobel Prize and the milestone of DNA, what make the DNA double helix structure discovery. But it was really interesting to also observe from him that he was also keen that how field can actually move and evolve from the genomics, from the DNA to the protein information at the proteome scale. So all those were really interesting interactions. I also had opportunity to share the stage with Ian Wilmot, who is the person who developed the clone Dolly. So some of these are really eye-opening experiences and good exposure. And eventually the trajectory moved on to the first draft of the human genome sequence, which got completed in 2002, 2003. And these were the Nature and Science papers, where now first time we knew that we have almost 25,000 were actually projected, 25,000 human gene. But now we know that there are around 20,000 human genes we have. But what is really interesting that different organisms have been sequenced from the bacteria to the fruit fly to the plant like Rhabdopsis thaliana to the C. elegans or yeast or human. All of them have almost similar number of genes, 17,000 to 25,000. At gene level, they are not very different. At even when you look at their specific molecular level, the biochemical level, lot of commonality you will find. What makes them so different is more changes at the functional protein level or at the metabolites level. So that's where the omics type of concepts comes when we try to understand what constitutes all the genes. Can we take a technologies and study all the genes of a given system, and that comes under the genome? Or we take all the proteins of a living system and try to understand that, and that comes under the proteome. And think about two simple examples. One is from the frog. You might have seen the tadpole larva, which is in the pond. And then you see a jumping frog, which is now on the land. This is the amphibian life cycle. The frog is amphibia. So now there is a very drastic difference into the aquatic life versus the land life, and it goes through these embryonic developmental stages. So the gene or the genome which constitutes this entire organism remains same in all the life cycle stage. From all of these stages, the genes which constitute this frog remain same. But that provides a blueprint information, a static information. From there, proteins are going to be functionally derived and going to make different type of proteins and more functional form of the protein information. And that's what is going to give its ability to move from this environment to move to the land environment. So from aquatic to land, it's like huge jump, and all that is only possible because of the proteins which are synthesized in this organism, and that is giving the functional information at the proteome level. A similar example, very elegant example for butterfly. If you look at larva, I'm sure you have seen many of these stages: larva, pupa, and adult form, and the adult butterfly. Butterfly looks so beautiful, but other stages do not look like so beautiful. All of them share the same genome, but they are having different protein constituents, and proteins are expressing dynamically at different stages. So that's where we are now more and more focusing, the entire scientific community is more and more focusing on understanding how the dynamic proteins are governing in our body. Can we find some cues, some clues, how they can be biomarker to understand a disease, how they can be biomarker to do a therapy or the treatment of a given disease like cancer. So what we learned today was essentially some really basic concepts, but I hope it is connecting you the dots, bringing you from the broad evolutionary concept to the origin of the cell, thinking about the cell, which is not just a living unit, it is much greater than the sum of looking at all its parts, it is deriving the functional information. Cell is going to divide to make the identical daughter cells, and if the cell cycle is not operating properly, it is going to dysregulate and show the phenomenon like the tumor formation. And the central dogma of life from the DNA, RNA, and proteins are really important to convey all the information, and that's actually being exploited, utilized, and investigated by the scientists working in the omics field, which they are now using different technologies to study DNA, different technologies to study RNA, different technologies to study proteins. And protein microarray is one of those important technology which has been utilized by all of, or the microarray is one of the technology which has been utilized by all of these three molecules. So DNA arrays to RNA arrays to the protein arrays, all type of arrays have been made, and people have tried to utilize that to understand the what is happening physiologically, what is happening molecular scale in the living system at the genome wide scale, to look at all 20,000, 30,000 genes at the microscopic slides. Can we print those molecules of DNA or RNA or proteins, and then try to understand different biological questions or clinical questions? That pretty much gives the the bird's eye view of some of the basics of the entire journey of central dogma, which is of course much more complex, as you can understand. But I think for this group, for this audience, for this course, probably it gives you some flavor of what we are talking, and why we are trying to emphasize to now learn more and more of the molecule at the omics scale, to look at all the molecules, all genes or all proteins or all the transcripts at the entire omics scale.